11 base pair barcode that identifies that vector, and that is flanked by conserved prime annealing sites that are the same for each vector. So what we now do is on each day of the infection in the mouse, we just take a drop of blood, extract the genomic DNA, the total genomic DNA from it, run a PCR to amplify all the barcodes, and then a second PCR to add sample-specific sequencing adapters, and we can multiplex many such samples on one next-generation sequencer run and count all the barcodes and lots of samples. So we can now, in one go, measure the growth rates of many, many mutants on one run of the sequencer. And that's something we've now done many times. So this is the kind of experimental design. We co-transfect 100 plasmids into chizons we get from an infected rat, because that really brings up transfection efficiency. We carry out 3 independent transfections in a Maxa4D multi-well format, and then we stick each of these populations into a different inbred mouse and monitor the infections every day by taking a drop of blood from the tail after we've treated with pyrimethamine to kill wild-type parasites. And from these samples every day, we amplify the barcodes and count them on a sequencer. And our phenotype that we measure is the relative growth rate. And we express that relative to a few control vectors that are the same in each transfection. And I'm going to show you now data from a genome scale data set that doesn't really cover the genome completely yet. So until now in the literature, 8% of P-Burger genes have been studied. A lot of the data I'm showing you is based on now a 48% analysis, and in a few weeks' time when we submit the data, we'll probably have 56% coverage of the genome. So the size of the experiment that has involved transfecting 2,571 vectors in 56 pools. Each vector is assigned to 2 different pools to account for any differences that might come from the genetic context of a particular pool. And we've infected 168 mice and measured 30,000 daily growth rate measurements. But it's something to know that the number of barcodes we actually counted, although there are millions and millions, is still much fewer than the number of cases of malaria each year. So it's been a big experiment. And we get lots of growth curves from that. In theory, how a mutant can behave within a pool is illustrated by this. And what you can see from it is that we've measured each vector. We've got measured each vector 6 times in different pools. For every type of mutant, the most robust measurement comes from a different day of the infection, right? So this mutant is gone by day 6, 7, 8, so you can't measure it anymore. So we had to come up with a good algorithm to measure the growth rates. And what we've done is we've modeled the variance in the data set for each day, and then we weight each measurement by the inverse of the variance and calculate 95% confidence intervals around a single growth rate measurement. So we just, from all this data for each gene, we in the first instance now just make a single growth rate call. Yeah, that is important to realize, because there may be mutants that change their growth behavior during the course of an infection, because an early infection provides a different environment from a late infection. But we're just ignoring that for the moment, because we think it will affect the minority of genes. So by definition now, we call an essential gene one where the 95% confidence interval on this relative growth rate that we now determine includes 0, where it includes relative growth rate of 1, same as controls. We call this a dispensable gene, and if it sits significantly between those two, it's a slow grower, or to the right, it's a fast-growing mutant. And, sorry, going the wrong way. And there's now a data viewer online that you can visit to look at the phenotype of your favorite mutant. You can Google for plasmogem and then follow the tab through to the phenotype data, and then you'll get this kind of display initially. And you can then go deeper into it and search for specific genes or upload gene lists, or you can download the whole data set and analyze it. But let us publish the whole data set first, please, before you publish it. But do feel free to use the data. So I want to give you a bit of an idea of the power of the data and the quality of the data. What we do is we plot the relative growth rate along this axis, so 1 is about here, 0 is here, against a measure of confidence that's derived from this 95% confidence interval. Just to give you an idea, if we take all values that have a confidence of 5 or above and then look for how these mutants have behaved in our different pools. Yeah, each mutant has been studied more than once, so most mutants have been studied more than once. When we look at the correlation, the reproducibility of all these values with confidence high of 5, we see that it's actually very, very good. Yeah, so our growth rate measurements are highly reproducible. It doesn't mean they're correct, but within what we're doing, they are reproducible. That's a good start. And you'll also see that the green dots here are the mutants that we call, the genes we call dispensable, and the red ones here are the ones we call essential. And the slow-growing mutants, which are of course very interesting for further study, are all the blue ones in the middle. They have a growth phenotype that we can now study right away to get at the function of these genes, an invasion or intracellular growth or egress or so. And this just to show there's only one really fast-growing mutant that we detected. That's actually shown here. And would anyone care to guess what that one is? Those are mutants where we have knocked out the transcription factor AP2G that commits parasites to gametocyte formation. In that we published independently a year or 2 years ago as the master regulator for sexual development. Yeah, had we not discovered it differently, we would have discovered it by now through this experiment. And it really shows there are no other master regulators of that, where the parasite can still recommit to asexual growth. And that's just an aside. So, the overall result is what I want to discuss with you now. What we find is actually quite surprising. So, we find that only a third of the genome don't give us a phenotype. Yeah? More than half of genes, or almost half of genes in this data set now, seem to be essential. And yeah, if you combine these slow growers and essentials, it means malaria parasites really need 2/3 of their genome to grow optimally during the blood stages, which is quite astonishing because there are all these other life cycle stages that we haven't even touched yet in our analysis. And this is again, Plasmodium. Now I'm lumping the slow growers and the essentials all together because that's what other studies have done. And then you see how unusual this is. If you look at essential genes in model organisms, you find there are far fewer in yeast grown in cultured human cells or in whole C. elegans grown under optimal conditions. But then you could argue, well, we've studied a pathogen in vivo, right? So it could be the whole in vivo thing, a much more complex environment. But if you look at Salmonella serotypes, typhi and Typhimurium combined here, those gram-negative bacteria actually have very large genomes. They've got 4,000 genes. It's not so different from malaria parasites. But to grow optimally in culture, they only need 295 genes, as was shown by Duncan Maskell and Julian Parkhill using the Atlas transposal screen. So you didn't have time to put those references on that. And then if they stick them into hosts to see which genes they additionally need to colonize them, you need an extra third of the genome to colonize the host. And you actually need different genes to colonize different hosts. Perhaps that's part of the answer, right? Malaria parasites are very specific. And then if you look at trips from the Ritzy screen, that forms were grown in blood stage forms, grown in culture. Again, only just a third of genes seems to have a phenotype. But this is again an in vitro experiment. So we think this is this high degree of essentiality is remarkable, and we need to look into that. Is it because we need to ask, is it true, right? I said our data are reproducible, but they might not be true. So let's look briefly into whether the data are true. First, we mapped the phenotypes onto the whole genome just to see whether we are missing out any parts of the genome that might be particularly enriched with essential genes. And red and green here give you the phenotypes around the chromosomes, and we don't see any particular pattern emerging there. We then ask whether genes we predict to be essential actually come back with a low growth rate. And that's the case. So ribosomal genes we pick up as being essential, whereas multi-gene family members that are exported and should be redundant because there are many of them, and most of them not expressed at any given time point, and they indeed can be knocked out very efficiently. So that seems to hold up. We then asked, what if we compare our data to published data, where people have taken a lot of effort to actually genotype their mutants, these 8% of the genome that was studied like that. And again, we see a good correlation. So this is a published phenotype. If that is essential, then we find the genes essential. Importantly, if that is dispensable, if people have found a viable mutant, then very rarely do we fail to knock that out. So that would suggest we do not overestimate essentials, at least not by more than these 5%. And you can predict that genes that are not expressed in the blood stages might be knockoutable, right? And so here we look at the sexual stages. We look at correlation between knockoutability and sexiness. Sorry, that will make it a dictionary mention. So we clustered all the genes in the genome by their expression patterns, and these clusters here, the red cluster here has peak expression in neokinase, which is the mosquito stage, and the green cluster here has peak expression in the gametocytes, the sexual precursor stages that develop in the mosquito. And those genes can really be knocked out very effectively, whereas genes that, if you look at genes that have alpha clusters that are well expressed in the asexual blood stages, the degree of essentiality goes up to 75% here. Yeah, so of genes that are expressed, really many have phenotypes when you knock them out. And that's in vivo. So that also adds up. And you can start now looking at metabolic pathways. I could give you a GOTERM enrichment figure that would stretch from, I don't know, here into the basement. And I'm not going to do that. You can do your own live GOTERM enrichment on our website by drawing circles around parts of these clouds of dots. So you can play with that and get your own idea of just looking at lipid metabolism to see whether things make sense here. And they do. So GPI anchor biosynthesis is known to be essential. That's highly enriched in essential genes. Let's see, where's type 2 fatty acid, fatty acid biosynthesis in the apicoplast is known to be largely redundant. These lipases, we haven't got an idea what they do, but they are redundant with some of them having interesting growth phenotypes. But then, you would perhaps not want to continue a drug program targeting single myelin and ceramide biosynthesis, and those programs exist. So generally, we can, although some uncertainty remains regarding the phenotype of individual genes, we can say that we can now associate many more metabolic pathways as being either essential or redundant, and that really is important for drug target validation. And yeah, this is showing the same thing for mitochondrial pathways. I'll skip over that in the interest of time. And you can ask questions like, are conserved plasmodium proteins, those that are just annotated as such because we haven't got a clue what they do and they have got no domains associated with them at all, are they any less important because we know nothing about them? And they are only slightly less important, as you can see here. Yeah? So most knockouts in these genes still have phenotypes in asexual blood stages. So they do something, and if we don't study them, we miss out on a lot of interesting biology that we might target. Although the actual strength of the growth phenotype is higher with functionally annotated genes. But remember, this contains all these ribosomal genes that are absolutely essential. So how does this all come about, and what does it mean? What does it really tell us about the parasite? My hypothesis that I want to put to you why so many essential genes are observed in Plasmodium compared to other eukaryotes that have been studied is that this reflects genome reduction during evolution. This is a model put forward by Arnaud Payne in a recent eLife paper where they describe the genome of Chromera, the sister taxon of all the parasitic apicomplexa, and they look at gene numbers on the one hand, but of course also on orthologous groups of genes. And they find, so these organisms still have 22,000 or 26,000 genes. That's similar to free-living plants. These ciliates as well have nearly 30,000 genes. And there has clearly been a reduction in the number of genes to the coccidia here, and then to the pyroplasms and malaria parasites here. And two main stages of gene loss have happened as the ancestral apicomplexan has acquired a parasitic lifestyle, with that many groups of orthologs being lost from the genome, and then massively again at the base of the hematozoa. So these, at this point, the parasites have acquired an intra-erythrocytic lifestyle. They have lost lots of their genes, and I would argue that's why so many genes are now essential. Yeah, that's not, that wasn't a foregone conclusion. We didn't expect to see that from published data that was out there. So, but stop, that can't be right, because we all know that Plasmodium is a prime example for genetic redundancy. Just look at Kirk. I mean, he's made a whole career out of studying VAR gene regulation, right? There are hundreds of genes that are exported by the parasite that are important for antigenic variation. There are these exported thick kinases. You all are interested in red blood cell invasion, and we know that there are all these alternative invasion pathways, right? And they might have co-evolved with the blood group system and are great examples of redundancy. And of course, yeah, these are very important pathways because they make developing vaccines so challenging. But I would argue what we're looking at there is the fluff, yeah, at the outside of the cell, what has co-evolved between the host and the parasite. And if we look inside, what we really are dealing with is a lean and mean Formula One engine of a core genome, yeah, driving the parasite around the life cycle. Yeah, and that's important because we can tell from that it will yield many, many more drug targets. And so perhaps that is why MMV have been so successful working with great collaborators in academia and industry to actually replenish the drug development pipeline and discover all these drug targets. Yeah? Because so many parts of that engine are really important. So I think I've shown you our data suggesting that a surprising 2/3 of all genes in the parasite are really important for a single life cycle stage. I remind you that all the data and the vectors we've used to create these data are freely available, so everybody can recreate the mutants and check out all the phenotypes, study them. I think this high essentiality reflects genomic reduction and evolution. And as a whole, the study thereby makes a contribution to drug target validation because on the one hand, we can measure the relative importance of biological pathways now. We have learned that conserved plasmodium proteins are important, but we also can predict that there will be plenty multi-stage targets for drug development still to be discovered. And if you are a more visual person, you just remember that. Okay? And these are the people involved in the work. The Plasmodium project is led by both myself and Julian, and Ellen is the staff scientist behind running it really on a day-to-day basis and keeping all the strings together, together with another staff scientist in computation biology, Frankie, who does all the database work and a lot of the computational analysis. Now Gareth here is very important because he's the man with the multi-channel pipette who has made these thousands of vectors. And Theo here is a PhD student from Julian's lab, has worked closely also with input from Leo Parks and junior faculty at the Institute in computational analysis to develop our algorithm for analysis. And I'll leave it at that. Thank you very much.
So one thing I noticed, I apologize, I was standing in the back watching the whole thing, but maybe I didn't hear you. In terms of the mutants, how many mutants go into each animal?
About 100.
So the reason I ask that question is if we just step out of the box and we say something is functional, it has a particular parasite growth rate, it's essential, it's non-essential, is it possible because of the system, and it's an in vivo system, that when you have a large number of mutants there, because what we know about immunity is the more complex what you're seeing, the more difficult it is to mount an immune response, right? However, there is some likely critical threshold, and if you throw something in and enough of it, you have a hyperimmune response. And that's going to mean that you would think some of these mutants would be summing together for the amount of growth rate that they get. And so it could go both ways. And I can't wrap my mind around it.
And we want to work with that. So that is, of course, a great phenotype to screen for, right? A mutant that behaves differently in a rat mouse or a skid mouse compared to the